Hello everyone, welcome back to Chinmoy's Biology channel. Today we are here back with a new episode of genetics and evolution. It is a very important topic. You have already studied this in class 10th as a lesson called heredity and evolution. So like that only we will be studying about the Mendel's laws of inheritance. Here we will deal with the deviations from Mendelism. So we are studying the incomplete dominance, codominance and all. We will be studying about the multiple alleles, inheritance of blood groups. So these are the main things we will be studying and dealing today. So stay tuned throughout the episode and you will be knowing many important things related to genetics and which should be known in your class for your competitive exam as well as for your 12th exam. So let's get started with today's episode of genetics and evolution. Now first we are starting with the lesson genetics and evolution, and this is as per NCERT book. So I'll be discussing with the notes and with the topic differently as we move throughout the lesson. So just let's get started with your CBSE NCERT book. So genetics and evolution. Okay, so it is unit 7. So chapter 4 is principles of inheritance and variation. So before that, there is an introduction. So the work of Mendel's and others who followed him, this all is the introductory part of genotype, phenotype and all. So I hope you all know what are these. Just go through it once. This is all about the history of Watson and Crick. So go through it once so that you understand about the theories and stories of this evolution. So this is all the theories it's being given here. So nothing to teach you right there. So just without wasting time, we will move on to this main part, that is principles of inheritance and variation. Okay, so here only we will be dealing with the main first part here, Mendel's laws of inheritance. So let's get started. Now first of all, we all are having a question that an elephant always gives birth to an elephant, baby elephant only, not any other animal. Why a mango seed forms only a mango plant, not any other plant? So why it is like that? So given that they do, are the offspring identical to the parents? So whatever the parents are, the kid should be identical to the parents. So do they show the differences in some of their characteristics? Have you ever wondered why siblings look similar to each other? So if you are having brothers and sisters, you will just see that you are mostly similar to each other, or even so different. Why you are not so similar? Why you are different? You are having different characters and all, though you are looking similar. So there are similarities and differences in our characters as well as in our external features, right? So these and several related questions are dealt with scientifically in a branch of biology called genetics. So this only we are going to study in details in 12th standard, right? So this subject of inheritance as well as variation of characters from parents to offsprings is the inheritance. So what is inheritance? Inheritance is a process by which characters are passed on from a parent to the progeny. Okay, so from a parent to their offspring, when the characters are passed on, this is called inheritance, and it is the basis of heredity. Now variation, what is variation? Variation is the degree by which the progeny differs from the parents. Suppose you are not looking similar to your father, to your mother, then it is a variation from the progeny, right? So it is a variation from the parents. Now humans knew from the early 18,000 to 10,000 BC, 1000 BC that one of the causes of variation was hidden in sexual reproduction, right? If they are sexually reproducing, there will be variation. So this was the thing. They exploited the variations that were naturally present in wild populations of plants and animals to selectively breed and select of organisms that possess desirable characters. What it means? That if they are being exploited, the variations that were naturally present in wild populations, if you are taking a wild variety of plants and animals, and you are selectively breeding the organisms or the plants, they possess certain desirable characters. If you are selecting the characters and breeding like that. So for example, through artificial selection and domestication from the ancestral wild cows, we have well-known Indian breeds. So if they have selected certain breeds of cows and they have mated them and sexual reproduction has happened, so then we got a well-known breed, that is Sahiwal cows in Punjab. We must, however, recognize though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of this phenomena, right? So they had very little idea that what was the reason behind this phenomena. They didn't know that. They knew only that if you are selectively selecting two wild variations of plant and wild variations of animals and you are breeding them, the crossbreed is being done, then only you will get certain variation in characters. They didn't know any scientific reason. So behind this, Mendel found out the scientific basis of this phenomena under this Mendel's laws of inheritance. So first we should know that who is Mendel. So then we will move on to who is Mendel. So we should study a bit about the scientist who has proposed the laws of dominance, the laws of segregation and the laws of independent assortment, what we are going to study today. So who was Gregor Johann Mendel? He was a biologist, he was a mathematician, he was a meteorologist, and he was staying in a monastery. He was born in a German-speaking family, and he was in a Silesian part of Austrian Empire, and which is today's Czech Republic. And he gained a posthumous recognition as a founder of modern science of genetics, right? So he stayed in a monastery where he worked with the pea plants. So he had huge fields where he used to perform experiments on pea plants, and Mendel worked with general 7 characteristics of pea plants, that is plant height. He also chose the pod shape and color, the seed shape, the color, the flower position and color. All that he chose were the easily visible characters. And why he chose pea plants? Because they were having many contrasting characters, and these contrasting characters were, during crossbreed and all, they were easy to handle, and carry out the experiments were very easy on pea plants itself. Okay, so for that only he did many experiments on pea plants only. So we will go with these experiments. We will start studying the experiments one after the another. So Mendel's laws of inheritance. So it was during the middle of 19th century that he head away with the understanding of inheritance, right? So Gregor Mendel conducted hybridization experiments on garden peas for 7 years. So this 7 years he dedicated fully to the pea experiments and proposed the laws of inheritance in living organisms. So during that investigations, inheritance, it was the first time that statistical analysis and mathematical logic was applied to the problems of biology. Before that, no statistical analysis was done, no mathematical logic was introduced into biology. It was just the theory part which people were dealing with. So what did he do? He had taken a large sampling size, which means that huge amount of plants were taken. So large sample size which have a greater credibility to the data that he collected. So more the sample size, it will be easier for you to do the statistical analysis, right? Also, the confirmation of his inferences from the experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance and rather than being unsubstantiated ideas. Okay, so generations after generations he continued these experiments. Then he tried to prove that, point out the general rules of inheritance rather than without any substantial unsubstantiated ideas, right? So he investigated characters of garden pea plant that were manifested as 2 opposite traits. So what are 2 opposite traits? Like we will see here, the 2 opposite traits in the Mendel's experiment one was dominant trait and one was recessive, right? So like in seed shape, if you are taking, dominant was round, recessive was wrinkled. Seed color, dominant was yellow and recessive was green. Flower color, dominant was violet, recessive was white. Pod shape, this was full and this was constricted. You can see the pod shape. Pod color, one was the dominant was green, the recessive was yellow. Flower position, the dominant was axial position and the recessive was terminal position. Okay. Now stem height, mostly they were tall and recessive was dwarf. So recessive means that they are carrying in the next generation, but they are not showing it phenotypically, that is physically it is not appearing in these plants, right? Now, Mendel investigated characters in garden pea plant that were manifested as opposite traits, which we have studied just now. Okay, so this allowed him to set up basic framework rules governing inheritance, right? Now, which was expanded on by later scientists to account for all the diverse natural observations and complexity inherent in them. So, he kept on working on these principles, and as the later scientists, to account for all the diverse natural observations. So there were several different natural observations and complexities inherent to them. Okay, so there were certain complexities. There were certain laws which came against this also. Deviation was also there, but hence few things he proved that they were very important. So Mendel conducted such artificial pollinations. What were they and what are the true breeding pea lines? We will learn this because he performed first the artificial pollination, cross-pollination experiments using several true breeding pea lines. So what is this? We will know first. True breeding pea lines. So what are true breeding pea lines? So between 1856 and 1863, we told in 7 years Mendel performed experiments. So hybridization experiments were performed on garden peas. So during that period, he chose that 7 pairs of contrast characteristics of peas and conducted some cross-pollination and artificial pollination on the pea lines that showed stable trait inheritance. That is, one trait or one characteristics is being carried from one generation to another, and this were underwent a continuous self-pollination. So such pea lines are called true breeding pea lines when self-pollination is done. That is, self-pollination is pollination within the same plant. So a plant having the male and the female features, and the pollination is done within itself. So it's not a cross-pollination, it's a self-pollination, and then only such pea lines are called true breeding pea lines. Now we will move to the experiment he has done. So now we know that what is a true breeding pea line. So this is a true breeding pea line, and a true breeding pea line is one that has undergone continuous self-pollination, that is one after the another generation. So this shows that the stable trait inheritance and expression of several generations. So the trait or the character is inherited stably from one generation to another, and there are several generations in this self-pollination process. So Mendel selected 14 true breeding pea plant varieties. So 14 breeding varieties he chose as pairs which were similar except for one character with contrasting trait. So all the characters were similar except only one character was different. Like suppose they were tall, another was dwarf. Other than that, all the other characters were similar. So some of the contrasting characters were selected, that like smooth seeds, wrinkled seeds, yellow color of the seeds, green color of the seeds, inflated pea pod, and constricted or yellow pea pods, tall plants or dwarf plants. So this is a full list of contrasting traits, right? So if stem height you are taking tall dwarf, flower color white violet, flower position axial terminal, pod shape inflated constricted, pod color green and yellow, seed shape round or wrinkled, seed color yellow or green. So these are the 7 pairs of contrasting characters. So now we will move on to inheritance of one gene, how he proved that. So let us take an example of one such hybridization experiment that was carried out by Mendel when he crossed a tall plant and a dwarf pea plant. So here the contrasting characters are tall and dwarf. He collected the seeds produced as a result of this cross and grew them to generate the plants of first hybrid generation. This generation is also called the first filial progeny or F1 generation. Okay, so what is filial generation or this filial progeny? This is the first hybrid generation. So if we want to define that, just let me explain that in another form. So what is first filial generation or F1 generation? Here it refers to the first filial generation, which means that these generations are the nomenclature given to subsequent sets of offsprings, right, from controlled or observed reproduction, right? So the initial generation is called P, or the parental generation. So, and the first set of offspring which are coming from this controlled or observed parents, they are the first filial generation or the F1 generation, right? So these are the F1 progeny or the first filial generation, and they can reproduce and create the F2 generation or the second filial generation, and so forth. So this process continues. So the first initial generation is the F1. After that they can reproduce F2 generation, and it continues like that. So this is the F1 generation. I think it is clear to all of you. So let's continue now. So this is what we have understood in the previous part, that is the first filial generation, some characters of the offspring were dwarf, and the character was not at all seen or expressed in F1 generation. It was not now expressed in the F2 generation, right? So in F2 generation, 3/4 of the plant were tall and 1/4 plant were short. So the tall and the dwarf traits were identical to their parental type only, but did not show up, okay? But they did not show any blending also. They didn't blend also. So that is, all offspring were either tall or dwarf. So none of them were in between height. So none of them were medium. So this is what he wanted to show us in his first experiment. So similarly, results were obtained with other traits also. He studied with other characters also, like purple flower, white flower, wrinkled seed, round seed. So in that, wrinkled was a recessive one and round was a dominant one. Like that, he continued several experiments. So he got a proportion of 3:2 in the second F2 generation, okay? And the contrasting traits did not show any blending either in F1 stage or in F2 stage. So that we should keep in mind that there was no blending there. Now, based on these observations, Mendel proposed that something was stably passed down unchanged from the parent to the offspring through the gametes over the successive generations. So these are called the factors, and now we call them as genes, okay? So genes, we keep on telling that he is having his father's genes or his mother's genes, because for that only he is genetically inherited that. So where come this came? So genes therefore are units of inheritance. So what are genes? Genes are units of inheritance, and they contain the information that is required to express a particular trait of an organism, whether he is that gene is dominant or not, whether the tall or the short gene is dominant. So these genes are expressing the information from the parent to the progeny, right? So genes which code a pair of contrasting traits are known as alleles. So genes which are coding for a pair of contrasting traits are called alleles, and they are slightly different from the same gene because they are coding for a pair of contrasting traits. So here he told that the alphabetical name he gave, for example, in case of the character for height, T, capital T is used for tall trait, that is dominant is always used with a capital alphabet, and small t is the dwarf or the recessive trait, right? So hence the plants and the pairs of allele for the height would be TT, 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 like this we will write, right? So how we are writing this? If it is tall, it will be, we will pair the alleles as T and T, like that only the crossing will happen, one from the father, one from the mother, so one from the male, one from the female. So like that only these are the codes for which the pair of contrasting traits are coming. So codes are written as T and T, okay? So Mendel also proposed that a true breeding plant, tall or dwarf variety, allelic pair of genes for height are identical or homozygous, okay? Homozygous means it will be TT, that is, it is carrying the same, both the allele are having the same factor. So TT, that is, both are dominant and tall, and both are, this dwarf is only dominant, this recessive is strong here, okay? So they are containing, the allelic pair of genes is homozygous, they are carrying the same code. Okay, they are having this, it is being
told that they are carrying the same code or identical code or genes, alleles they are carrying. Okay, so allelic pair of genes of height is T and T. So we will now.
So what are alleles? Alleles, if we want to discuss in very simple language, they are a pair of genes that occupy a specific location on a particular chromosome. So these are chromosomes. They are a pair of genes which are located on a specific location on a particular chromosome. So this is a chromosome and alleles are being located on a particular chromosome and they control the same trait. Okay, so alleles, they may be in pairs or in multiple forms of alleles which affect the specific trait of the offspring, right? They may be heterozygous and they may be homozygous alleles. So if they are heterozygous, so one will be denoted as capital A, another one is denoted as small a. So it is AA. This chromosome is having AA. So all the alleles are A, capital A and small a. So one is dominant, another one is recessive, and here this one is recessive and this one is dominant, right? This is a heterozygous allele and this is homozygous allele. Here these two are AA and these two are AA. So this here you can say that if this is carrying the trait of tall plant, this will be carrying that of a dwarf plant, like that only. This is, I am an example, I am giving an example to you all. So this is just an example to understand what are alleles and how they are located. So this is a chromosome, they are located in this part as a particular, at a particular location, in a specific location, and they control the same trait. Okay, so whether it is tall, whether it is dwarf, whether it is round, whether it is wrinkled, so they are being responsible for the specific trait of the offspring, right? So now we will move on to what we were studying in the book, that how these alleles are taking or transferring the characters from the parent to the progeny, and how this dominance and recessive characters are carried out, and what is a phenotype and genotype. So that we'll be discussing now.
So we have spoken clearly about the alleles in the last slide. So now TT and TT are called the genotype, as they are carrying the genetic characters genetically of the plant, while the descriptive terms like tall and dwarf are the phenotype. So the genotype is the genetic form of the plant, whereas the descriptive plant which is visible to us, the tall and the dwarf, are phenotype, right? Now, as Mendel found that phenotype in the F1 heterozygous TT to be exactly like that plant TT in appearance, he proposed that in a pair of dissimilar factors, one dominating and the other is recessive factor, right? Hence he called one is dominant factor and other is recessive factor. So they are genetically different, right? So TT and TT, but phenotypically they are looking same. So both are tall plant. You can't differentiate when you are looking at these plants. They are both looking tall, but genetically they are different. One is TT and another is TT, but phenotypically they are same. So he told that this one is the dominant one factor and this one is the recessive factor, right? So T is standing for tallness, capital T, whereas the small t is standing for dwarfness, right? So this is recessive. See, he observed identical characters, trait pairs that is studied. So it is convenient and logical to use capital letter and lower case of an alphabet symbol to remember the dominance and recessiveness, right? So T and T, we are not writing D for dwarf. You are writing T for tall and small t for the dwarf plant. Okay, so in case of homozygous, we will write TT and TT, and it is heterozygote, it will write capital T and small t, right? Since the TT plant heterozygous for genes controlling one character, it is a monohybrid, and hence the cross between both the capital T and T homozygous alleles, this one and small t is a monohybrid cross. Now we will concentrate in this monohybrid cross in the next class, where we will go to details about this monohybrid cross as well as the dihybrid cross, and we will read that in details, and we will read this also with the detailed description in the next class. We will read also about the Punnett square, and also you need to subscribe to my channel. Stay tuned with all the episodes which I'll be posting, because if you are not subscribing, you'll not get the updates whenever I'm posting, and you need to continue this lesson in a systematic manner. So stay tuned, stay updated. Please do subscribe to my channel and share it with your friends, and please don't skip in the middle. So as of today, please do go through the full topic once again, refer your textbook which you are going through, and wait for the next episode which I'll be bringing very soon with the monohybrid cross and the dihybrid cross and the description of all these things. Thank you for watching my video with full patience. Thanks a lot.